Well, thank you very much. Thanks to Professor Andrea for the invitation. I'm really honored to be here and talking with such a great group. I'm going to talk a little bit about the work we've been doing on the human microbiome. And I didn't start out really as a microbiologist, and I certainly didn't start out doing work in the clinic. In fact, when I got to MIT, I said that there were enough people working on medical applications that I was going to work on microbial ecology, and I was going to avoid working on anything biomedically related. Oh, thank you. Oh, perfect. Thank you. I'll take that view. Then I can walk around. I specifically said I would avoid working on anything medically related. But around the time of the Human Microbiome Project, a colleague came to me, and my background is in math and computer science as well as biology. And he said, Eric, we need you to help out a little bit on the statistics for, how do we tell if a bacteria is overrepresented in sick people and underrepresented in healthy people, or the other way around? How do we do that statistically? And I thought it was a really interesting challenge, and I thought I wouldn't get too sucked into the biomedical side of things if I was just doing something as innocuous as statistics. But that was about the time that the Human Microbiome Project was getting started. I guess the rest is kind of history. My lab is now completely focused on biomedical uses for the microbiome. We got funded by a really great philanthropist in the Boston area, the Rasmussen Family Foundation, and that's allowed us to create the Center for Microbiome Informatics and Therapeutics. So a lot of our work now is based on trying to use microbes as therapeutics, so not really thinking to traditional small molecule drug development, but actually using the bacteria in our guts to cure disease. So this is a graph I like to show, and if you've ever seen a talk by me, you kind of already know what this is. This is how I got involved in human microbiome work in 2009. My graduate student and I decided to launch a study where we would track absolutely everything that we could think might interact with the human microbiome. And so we tracked our sleep habits, we tracked every morsel of food that we ate, okay? We tracked sickness, we tracked over 300 different variables. If we got up in the middle of the night, for just 30 minutes, we would track that. We'd throw it into our phones. In fact, we tracked how much time we spent tracking. I used about 8 hours a week just thumbing things into my phone, keeping track of all. And this is the pattern that came out. So this is how the bacteria behaved over the course of a year in me. And this is how the bacteria behaved in my graduate student, Lawrence, who's now an associate professor over at Duke. And the one thing that you can take away just by glancing at this is that I can pick any day from my time series. You would know it's uniquely from me. It has a very different signature from Lawrence's. And it wouldn't matter if I took a time point at the beginning of the time series or at the end of it. It has a unique signature. And so it also means that the microbiome is very stable over time. But we did find that some of the factors that we spent all this time tracking also correlated with the microbiome, mostly diet, and in particular, mostly fiber. This is another view of the same data. So this is my microbiome in the year 2009. And again, on the x-axis, you can see time. And on the y-axis now, each row is a different bacterium. And I've scaled them so you can see more bacteria this way. In the previous plot, you could only see about 10, because you only see the biggest ones, and the smallest ones get, kind of obscured from view. But here you can see many more bacteria. So the bacteria are all the different rows, and of course the x-axis is time. And the warmer colors, the reds, means there's more of that bacteria on that day, and the blue colors indicate there's less of that bacteria you can see the main signal here happened about 3/4 of the way through the study, where I went out with a colleague for breakfast, and I ordered some French toast. It was recommended as the chef's special, but it was a little undercooked, and I ended up getting a salmonella infection because of it. So, of course, I went to the urgent care and told them I think I had food poisoning. They said, of course you don't. Everybody thinks they have food poisoning, right? But you certainly don't have food poisoning, and they gave me some IV fluids and went home. But later, when we got it on the DNA sequencer, we found salmonella. So the doctor was wrong. I did have food poisoning, and we had DNA proof of it. But the doctor is also the chief of our ethics board, so we don't like to bring that up and present this at MIT. So what happened next was really kind of surprising, though. So I didn't get any antibiotics, which was kind of the silver lining there, is we got to see the course of the salmonella infection sort of undisturbed by antibiotics. And what salmonella does is part of its pathogenicity mechanism is it doesn't avoid the sort of radar of the immune system. It intentionally induces inflammation, and it intentionally causes the immune system to wipe out a lot of its competitors, and thereby it takes over a lot of the biomass of the gut is just salmonella for a short period, and of course then that can infect other people as well. So we have a week or two of salmonella, and then following that week, we can see that some bacteria are really abundant that were never abundant before, and some bacteria that were abundant before are completely gone. Okay? A few of those bacteria that were driven away by the salmonella came back, but the majority of them basically went extinct, at least within my gut. They never came back. And so we can look, and this is a view, this is sort of the tree of life of organisms that live in my gut, okay? And what you can see here are the blue species, and these were the species that were present before I got salmonella, and the orange are the species that replaced them after salmonella. And so you can see for every clade, for every group of organisms that was abundant before, there are clades of organisms that were abundant later that were very, genetically very similar. And so that's why on the first slide you didn't really notice there was any big change from the beginning to the end of the year of my microbiome, because these organisms were replaced with very similar organisms. And all of them were a special group within the firmicutes, okay, within one region of the tree of life, whereas some of these other regions were not very easily replaced. They were able to kind of weather the storm, and they didn't really disappear after the salmonella. Okay, so that got us thinking that if during the course of natural events we can sort of reboot and reprogram the gut microbiome, maybe we can start to think about engineering the microbiome and putting bacteria there that we think are going to be beneficial for health. And part of that means, well, we need to know sort of what healthy means in most cases, except for one case here. This is a recurrent Clostridium difficile infection. So this is the most common hospital-acquired gut infection in North America. And what generally happens is we have patients, and they come to the hospital, and we can think of this, evergreen forest here as a healthy gut ecosystem. We give them antibiotics that increases the probability that they're going to get a gut infection afterwards, because we can think of it as taking this healthy gut ecosystem and then clear-cutting the forest, and then we can get invasive species. And one of these invasive species in the gut is Clostridium difficile. So then the patient comes back to the hospital. Now they have a Clostridium difficile infection, so they have a gut infection, so what do we do? Well, of course, they have a gut infection, so we give them antibiotics. and we bring them back here, they can get infected again. And so, of course, we give them antibiotics, and we go through the cycle about 3 times. And on the 3rd time, then they're classified as recurrent C. difficile. And then we have another option. And the other option is, after they've taken these antibiotics, instead of letting C. difficile get a foothold, what if we were to take this healthy microbial ecosystem, take the bacteria from a healthy individual, and put it on top of this background where we've just given the patients antibiotics. And so that's our fecal microbiome transplant, and the idea is that we go from this clear-cut form all the way back to a fully functional, healthy ecosystem. And this has a 90% cure rate, even in patients who have failed standard antibiotic therapy 3 times in a row. And so in my lab, sort of at the end of 2013, we started up a nonprofit where we prepare these samples because doctors didn't really know how to isolate these gut bacteria on their own. And we would mail them to doctors who wanted to perform this procedure. The organization has grown quite a bit since then, and so now we've treated over 25,000 patients within the U.S. In fact, more than 50% of patients now who get recurrent Clostridium difficile, instead of getting antibiotics, they'll get a fecal transplant from our nonprofit organization. And I'll talk a little bit about that Open Biome because in the process of building that organization, we've had access to some really great resources that have allowed us to do some cutting-edge science as well. So I'll talk about one study. This was a study we did at Mass General Hospital, initiated by Libby Holman, Alon Youngster, and Jenny Sauck, and then in collaboration with Dirk Jeebers and my co-director at the Microbiome Center, Ron McXavier, who's head of GI medicine over at Mass General Hospital. And so what we did is we had 4 donors. Okay, so this is the blue folks on the left who donated fecal samples to 20 patients that had recurrent C. difficile infections. About 90% of those patients got better, so 18 out of 20. And what we did is we did sequencing before and after the fecal transplant. So here what you see, and I'm not showing you all the Latin names, it would just be kind of gobbledygook on the top here, but each column in this graph is a different bacteria, and so white means that bacteria is absent in that particular sample, and dark blue means there was a lot of that bacteria in that sample. So this is our patient. It's a C. diff patient before treatment. This is our donor. Okay, so you can see the donor shares some strains in common with the patient, but not many. Okay, and then what we wanted to do is to be able to predict what will happen if I take this patient and give them a fecal transplant with this donor. What's it going to look like? We thought about it. We kind of looked at this graph and we said, well, it's not just like adding the top and the bottom to get the answer. It's not an AND gate or an OR gate or any of those things. And so Chris Smiley, who was a graduate student in my group, decided to see if he could build an algorithm to predict what this would look like using some machine learning techniques. And so this was his predicted algorithm, and it actually did quite well at predicting not only whether a bacteria would engraft, as we say, it would colonize the patient after the fecal transplant, but it would also predict the abundance of the organism if it did engraft. So that was great, and the next thing that Chris did is we wanted to know, so what I was just showing you is information at the species level, and we wanted to get down to really fine resolution. We wanted to look at the strain level because a particular species might be there in the form of 3 or 4 or 5 completely different lineages, and so we wanted to know how many strains for each one. And we had metagenomics, and so this turned out to be an interesting mathematical puzzle. If we just had a room full of people who didn't share any strains in common, the best we could say statistically is how many different strains each person had. Okay. However, if in this context, we're taking strains from one person and we're putting them in up to 12 different recipients. And because a strain might be highly abundant in one person and lowly abundant in another person and absent in a third person, we could use those patterns to not only figure out how many strains there were, but to actually figure out the genotype, the complete genome sequence of each of those strains. And out of this metagenomic data, which is usually very hard to assemble into individual genomes. So strain finder was the algorithm we used to do that. I won't go into too much detail, but here's just an example. So here's a group of genomes. The black genomes on the tree are the actual genome sequence, and the reds are the predicted strain from strain finder. So you can see in this situation, it actually does quite well. And this is with 16 strains and 16 samples, okay, and 1,000 SNPs. Okay, so what does it look like in our fecal transplant case? Well, in some of the cases, we have a particular species in the donor, and we don't have that species in the patient. And so that's what happened here. This is donor number 1, and the species is Ruminococcus. And donor number 1 has 2 different strains of Ruminococcus as predicted by Chris's algorithm. The blue strain is more abundant than the green strain. And after the fecal transplant, these samples are about 2 weeks apart. Okay? So after the fecal transplant, even though the patient did not have Ruminococcus initially, the patient has both strains, the green one and the blue one from the donor, and they stay fairly stable over time. Here's another donor that had the same species, but 4 completely different strains. And so this donor was used to treat a different patient, and you can see that all 4 strains made it into that patient, and they stuck around for the entire length of the study. This is another bacterium, Faecalibacterium prausnitzii, where our donor had 4 strains, and the patient picked up all 4 strains after the fecal transplant, but then something unusual happened, and the patient picked up this red genotype here, something that wasn't in the donor and wasn't in any of the other patients in the study. And we saw this happen over and over again, and we called these environmental strains because they somehow came from the environment. So this patient never had F. prausnitzii before, but after getting a fecal transplant and after getting just a critical mass of F. prausnitzii from the donor, the patient starts to pick up unique strains. So there was something about having some F. prausnitzii there allowed that patient to get even more F. prausnitzii, perhaps from the patient's diet, or maybe it was there in very, very low level in the appendix or hiding out somewhere. But it wasn't until they got the fecal transplant and got some F. prau that they were able to, those strains were able to emerge. This is another example of a case now where the donor has the species and the recipient has the species. So totally different strains. We wanted to know what will happen if the patient already has that species and we give them a fecal transplant. Will they keep the species they have, or will new strains outcompete them and push them out of their niche? And so we saw both cases. In this case, after the treatment, the patient looked just like they did before, mostly like they did before. And in this patient, after the treatment, they looked very much like the donor. So we saw both cases can actually occur. In general, what we saw is that when patients got strains from the donor, even if the donor had a lot of different strains, the patient would pick up all of those strains. It was an all or nothing phenomenon. In most cases, it's worth pointing out, in most cases, the patient didn't pick up any strains from the donor, and 70% of the species just didn't engraft. But if they did engraft, all the different strain genotypes engrafted. This, what I'm showing you here in blue, is how many strains came into the patient that were not in the donor or the patient before the study, these so-called environmental strains. You can see it's quite a large fraction in most of our fecal transplants end up being strains that were not even, that were not from the donor or the patient. Okay. So that gets closer to this idea where we can engineer the microbiome and engineer it to maybe optimize better health. And it's certainly something that we're actively involved in, using fecal transplants to make folks healthy again. But where we'd really like to go is synthetic microbial therapeutics. So we'd like to culture these bacteria in isolation, pure bacteria, and make something that looks a lot more like a drug. Or we could call it a probiotic if you want to call it that. But a probiotic tailored to a particular disease. And so we took the organization Open Biome, which does the fecal transplants, and we looked at all the samples in the freezer, and we found that we had quite a large number of samples to choose from, and we wanted to characterize them in great depth. So we tried to isolate as many strains as we could. We did metagenomics on a number of strains. We did 16S surveys, and then we also did metabolite profiling. And so we were able to get a number of samples from about 86 different donors here. So not patients, healthy subjects. These were the subjects that are being used to give the healthy bacteria to our patients. And for a number of these, we got very long time series, in some cases over 2 years worth. So we were able to isolate these. I think we have about 7,000 isolates now. So we isolate, get the complete genomes, sequence the complete genomes, and then store them in the freezer so we can do more analysis on them later. And this is our pattern. So amongst the major groups, the dark kind of shows how much of that diversity we've already captured and we already have in the freezer for 11 different individuals who live in the Boston area. Okay, so you can see for the Bacteroides and the Firmicutes and most of the different groups, most of the biodiversity there we've cataloged it, we've sequenced their complete genomes. And this includes some species that were previously thought to be quite hard to isolate in culture, like Akkermansia. There had only been one human isolated strain in culture. It's an important part of the mucosal microbiome. And we've got about 60 isolates now from 5 different people. Tricibacter is another important component where there was previously one human strain. Now we have over 500 of these spore-forming organisms. So we looked deeply at a few of these already, and it's kind of ongoing work. But what we found, and this is Bacteroides fragilis. These are 12 different donors that we isolated B. fragilis from, and we isolated about 30 strains from each. And all of these groups formed a single clade. So what that means is all of the diversity in each person results from a single bacterium inoculating that person. And I'll give you an estimate of how long that bacteria has been dominated in each person's gut in a minute. But B. fragilis was kind of interesting in that most of the evolution we see happened within a person. And so this is work done by Kenny Lieberman, who's now an assistant professor also at MIT, and Shurja Zhao, who's a graduate student in my lab. And so we can look at each of these lineages and kind of see the distance to the most recent common ancestor, and in most cases it's under about 10. Except for one lineage where we had a hypermutator that had up to 72 SNPs. And we know it's a hypermutator because we can look at the mutation frequency, and we see this excess in transition versus transversion, which is pretty characteristic of a hypermutator. And in fact, the particular kind of transition is highly skewed as well. But what's more interesting, to me at least, is not the hypermutations, but those mutations that are actually under selection, because I've already told you that each of these lineages resulted from a single clonal bacteria entering that individual. and then all of the diversity happened within that person's gut. And so if we have evolution, what we're really interested in is how is this bacteria adapting to a single person? Presumably it came from another person, so it's already adapted to humans. But if I eat a bacteria, if I take a probiotic, how does it adapt to being in my particular gut rather than your gut, right? That's a fascinating question. Can we identify these? And of course, it's hard in bacteria because they do reproduce clonally. We don't have a lot of population genetic tools that we can use to identify positive selection. So what we thought is, if we see multiple non-synonymous mutations in a single gene or in a pathway of genes, that tells us that something might be going on. And so we looked and we said, if we start to see multiple mutations in a single gene, whether it's in the same lineage or it's two different lineages in parallel, is the number of non-synonymous versus synonymous substitutions going to be much higher? And when we do that, we find, well, if we look across lineages, the DNBS ratio is quite low. It's what we expect for most genes. However, if we see multiple hits, it's anywhere from a ratio of 3 to a ratio of about 16. Okay, and in fact, we saw in these genes that had multiple hits, they were about 6-fold enriched in membrane proteins compared to intracellular proteins. And so this gave us a lot of confidence that the strongest hits really were under adaptive evolution. And so what are these genes? Well, a lot of them had to do with capsule polysaccharide biosynthesis, which makes a lot of sense, and transcriptional regulation and some carbohydrate usage. But what we saw, at least for B. fragilis, over and over and over again, we saw things in the sus-C, sus-D family. And these are major membrane proteins that are used in carbohydrate attachment and carbohydrate uptake. And what we saw is that mutations tended to occur in the hinge region and also the interface between sus-C, sus-D, implying that they had some effect on gene function. And so we haven't really sussed out exactly what the fitness effect of these genes are. We've nailed it down to a couple of possibilities. One is that there's two lineages that harbor different phages, and we think that the phages might be, they're using the phage to attack each other individually. So we know that, if you eat cornstarch versus potato starch, different variants of sus-C and sus-D are needed to access those sugars, even though chemically they're both starches. So here in donor 44, we can go back to our metagenomics, and so we have all of our strains, and we can build this phylogenetic tree, and it's a perfect parsimony tree. So we know exactly where the root is and what the phylogeny is. And we can look at where these mutations occurred and trace them over time in our metagenomics. So we have metagenomics every day for this donor, or let's say most days for this donor, over the course of about 2 years. And so what we can actually see is we can see mutations arise, like this blue one. We can see the mutation arise here, and then we can watch it as it's selected. And we can even go in and do things like directly measure the selection coefficient of this new mutation. Here's just another example. And so sometimes we see new mutations arise and then disappear, and sometimes we see new mutations arise and basically go to fixation. And so now we can document this in microbes in their sort of natural environment using both complete genome sequences and metagenomics. This is work that we did a while back, and this is Chris Smiley again, who wrote the Strain Finder program. Mark Smith, who started the nonprofit organization Open Biome, and Jonathan Friedman, who's now an assistant professor at Hebrew University. We got excited about this because there's an interesting story. It's a story in Nature about carbohydrate active enzymes that had been horizontally transferred twice. So these are the basically the enzymes that degrade this porphyrin polysaccharide that's in these nori sushi wrappers. Okay, so they're highly sulfonated, and most organisms can't access those polysaccharides because of those modifications. But the algae that make them, of course, they have the degradation algorithms. Well, there were some marine bacteria that basically stole a copy from the algae, and then they made a living by just living near the algae and then eating the polysaccharide from the algae. At some point, probably a Japanese individual ate one of those pieces of seaweed that had the organisms that had already stolen the gene from the algae on it, and the bacteria in their guts stole the gene from the bacteria that stole it from the algae. And so now if you look in a North American gut, we have a lot of comprehensive metagenomic surveys. You won't find this gene at all. But if you look at a Japanese metagenome, you'll find it's ubiquitous. Almost everybody has organisms that have these genes, and they're in the Bacteroides. So we have a lot of Bacteroides in North America. They have a lot of Bacteroides in Japan, but these mobile genes are quite different, and they're allowing us to access different substrates. So in 2012, we published this. It was the first ever map of horizontal transfer, and we did not only the human microbiome, but also these non-human environments. And we always saw that there was more horizontal transfer occurring amongst bacteria that came from the same environment, but there really was a very high level of transfer from things that came from the human body. And in particular, 2 organisms from the gut were highly likely to share. So were 2 organisms from the mouth, and that was more likely than an organism from the gut sharing with an organism from the mouth. So the more precisely the habitat was determined, the more likely they were to share genes. And these are, importantly, bacteria that were isolated just using the organism gen bank at the time. So all these bacteria were from different people. It's just that they were from the same body compartment. So we got interested in kind of taking this out into the field, and so Alana Brito was a postdoc and now an assistant professor at Cornell, wrote a proposal to go out to Fiji and start collecting these so we could look at the differences in their microbiome, and importantly, how bacteria were being shared across people and how genes were being shared across bacteria. And so she wrote a proposal to go out and basically collect the same type of data they were collecting at the time for the Human Microbiome Project, but also figure out all the social networks, get people's medical histories, figure out where their water supply was, their food supply, GPS coordinates of their home, figure out everyone's family tree, and collect the kind of metadata that the Human Microbiome Project didn't have. Well, the NIH looked at that and said, well, you're proposing to do even more than the Human Microbiome Project, which we already spent $140 million on. So there's no way that a single postdoc could do this on her own. And so they said, scale your project way, way down so a single postdoc can do it, and then we'll think about funding you next year. Instead of doing that, she flew out to Fiji on her personal credit card and collected thousands of samples, which she brought back to the U.S. And we wrote a proposal after the fact to the Broad Institute, who had some money at the end of the year that they had to spend down on one of their grants, and decided to very generously fund complete metagenome sequencing for all of Alana's samples. And so what she found was the Fiji metagenomes all clustered together very far apart from the HMP, US genomes, and the different Fijian villages were all sort of very similar. And when we looked closer and we looked at the particular gene families, what we found is that there's a number of gene families that were universal. Okay, so red is Fiji, blue is US, Human Microbiome Project. And so there were universal genes, but then there were a few genes that were mostly present in the US, and there were a lot of genes that were really only present in Fiji. And when we started to look at those, we saw a lot of glycoside hydrolases and a lot of genes that were relevant to diet. We saw some differences in things like antibiotics. And importantly, when we started to look at the gene level, we actually saw differences between different villages. So I just showed you on the sort of scatter plot that at an overall community level, they have roughly the same organisms from village to village, but some of the genes were really unique to a particular village. And we don't really know why that is yet, but we suspect it could have things to do like diet, or maybe just that these genes are being transmitted very, very quickly, and people travel in between these villages very rarely. Probably don't have time to talk about this story, but after we published this work, I got a call from what I thought was a really bright postdoc at Tel Aviv University, and she called me up and demanded to have a Skype with me. I said, okay. And she said, you know, Eric, I just read your 2012 paper in Nature on horizontal transfer, and I want to tell you that you got everything wrong. And the reason you got it wrong is because in order for us to identify a gene as being horizontally transferred, we said it had to be 500 base pairs long, and it had to be roughly 99 to 100% identical in two totally different species. She's like, all of the interesting things are happening in those pieces of DNA that are less than 500 base pairs long. And I'm going to come out to your lab and prove it to you. And so that's what she did. What she found is that this regulatory information in between genes that are not horizontally transferred, there was a huge amount of horizontal transfer of promoters and transcription factor binding sites, and so all this regulatory information. And ended up publishing that in 2014, and she ended up winning the Casarelli Prize for the best biomedical paper in PNAS that year. Okay, so I talked a little about horizontal transfer. How does that relate to these organisms that we've studied over time? Well, here's a lineage here, and you can see in light blue are the isolates that we got on day 1, and in red are the isolates we got a year later. And so we can see this mobile element is only showing up in the second group, and it forms a clade. And so we think this is horizontal acquisition during the course of our study of this particular gene. We can go back on day 0, and we find that it's not in any of the isolates, but it's in the metagenome. So it's in other groups. And we've since gone and we've actually found it within our complete genome sequences in Bacteroides vulgaris. And so we think we can say that it's been transferred from B. vulgaris into B. fragilis, and it happened during the course of our study. So we can actually see this horizontal transfer happening over time. We've looked at other organisms like E. coli, and here the dynamics are very different. So this is day 0. This is the first day of that we sampled. This is about half a year later. We see 2 different groups. One is quite similar to the original, but then about a year later, we see that they've all been totally replaced, and the diversity is now nearly clonal. And then on the final time point, they've been replaced again. And there's 2 different groups, both of which are nearly clonal. So we see that in Bacteroides, we build years or even decades worth of diversity, whereas in something like E. coli, the diversity is constantly being purged, and so these different organisms have different lifestyles. Maybe the last thing I'll tell you about is how we're looking at the morphology of these organisms and how that's evolving over time. And so I showed you that they can pick up genes maybe even in the course of your lifetime. They're picking up genes to adapt to your personal diet and antibiotics. And they can also change form. So we know a lot of these bacteria form endospores. And so Sean Kearney, who's a terrific graduate student in my lab, has taken the whole community, treated it, leaving only the endospores intact, and then he compares, using 16S sequencing, the whole community versus the endospore fraction. And he did this in 24 different individuals, and then he did it in one individual over about a month's worth of time. And so you can see in the beginning we have a lot of diversity. This is the bulk community. And then after treating and enriching for endospores, we really only have Clostridia, actinobacteria. I won't even try to pronounce this one, but it's a firmicute, one of the firmicutes, and then the bacilli. And so now I'll go back to these data. So this is, again, my microbiome, and you can see the bacteria that came before my salmonella infection, okay, are highly blended here. Only a few of them are spore formers. And then after the salmonella infection, you can see that they're dominated by, the ones that came up are dominated by the spore formers. So this part of the phylogenetic tree that I drew your attention to earlier, this is precisely the part of the tree that forms spores. And so we can see that these spore formers, it's easy for them to transmit because they were able to re-inoculate me within days of my infection, but they're also the shortest lived. And so we think there's a trade-off between being transmissible across many people and being persistent in one person, like the Bacteroides. We know these spores play a role in infectious disease, so back to our recurrent C. difficile infection, we see that our healthy controls have a lot of spore formers. People who get Clostridium difficile the first time have relatively fewer of these spore formers, and our recurrent C. diff patients have very few spore-forming organisms. And so the sort of level of spore formers in the gut is indicative of this disease. And finally, to just reinforce this notion that this is the more malleable, variable portion of the microbiome, we can see that in the case where I said sometimes we do a fecal transplant and the patient stays like they were before, sometimes the patient ends up looking like the donor. Well, if we're looking at spores, then we see the patient ends up looking like the donor. And if we're looking at non-spores, then we see it's much more likely that the patient is going to retain their own strains. And so, you know, almost the only cases where the patient keeps the strains they have is when those strains are something like the Bacteroides and not a spore former. So with that, I'll thank you very much for your attention, and I'd be happy to take a question or two if you have time.
Thank you. Regarding your salmonella infection, how long it took for you to get back your diversity? If that ever happened.
Yeah, so it was very, very quick. It was just a few days. Like as soon as the salmonella went away, the other organisms were there. So it was very, very quick. And then the surprising thing about that is that it was stable. You know, it was, you know, very rapid uptake of, you know, a couple hundred organisms, and then you know, very, very slow turnover after that to normal.
Very interesting. And I was thinking about the immune system, and especially the gut immune system puzzle. And my question is if you investigate the background of that, at least the puzzle immune systems from the donor and also for the receptors, and if it would be information that would help to predict the outcome of the transformation.
Yeah, that's a great question. So there's these differences between people, and we don't really know what it is. It could be diet, it could be immune system, it could be other factors, or some combination of those, right? We don't really know 100%. I can give you some anecdotal stories. So one thing that we've done is we've taken mice, and we've taken their gut bacteria and their lymphocytes, and then we put them into mice, new mice, which are RAG deficient, so they don't have lymphocytes of their own, and we try to see if you get the right bacteria but the wrong lymphocytes, does something unusual happen, things like that, and that hasn't really been as clear a signal as you would think. We're just now starting to look at which bacteria are labeled by IgAs, because we think that can be pretty important. And so one thing we've done is we've taken that gene, or that organism with the gene that allows it to eat sushi, and we put that into mice, okay? And then we've taken the mice and we've either fed them seaweed or not. And what we find is that if we feed them seaweed, we can get that organism to engraft and stay there. And if we don't feed the seaweed, then the organism can't engraft. Later, if we take the seaweed away for a few months and then we go back and then we feed them seaweed again, the mice that didn't see it the first time will respond to the seaweed and the organism will engraft. Some of the mice that got it the first time, they won't respond. And when we look, those organisms are now being targeted by IgAs. So one thing we know is if you create too much of a single organism, then they can start getting targeted by the immune system. Then it would be very hard to put that in. I think it's a super exciting field. Like nobody is doing a lot of T cell receptor sequencing and IgA sequencing along with microbiome right now. So it's like hugely unexplored.
I was wondering if you could do something more simple, like genotyping the MHC of the H donor H receptor. So maybe you have a correlation, like people that are TQ2.2, they favor some bacterial species from people that are DR7, for example.
Yeah. So there better. I don't think that's going to work without like a very large population sample. Maybe if you were to recruit a few like key MHC types, you could do it. There have been a number of different GWAS studies, and I assume that these would probably show up there. They found a few things, but really only like a handful, like one or two genes, and that's with thousands of people already. So the other thing is things could be like really confounded by diet. So doing everything with this, ignoring the diet component is probably also tricky.
Do you think the immune response could explain why some strains get expanded in the recipient compared to the diversity that you see in the donors? And if you can, if that's possible, to check the antigenicity of the variants in the donor and see if there's a correlation with the strains that won the race in the donor.
So I think it's possible, but remember, using our algorithm, which doesn't have any information on the immune type of the host, we already can predict about 86% of that variance, and we can also predict how abundant it's going to be. So I think the abundance is determined by things like, if you're a bacteria that specializes in omega-3 fatty acids versus a bacteria that specializes in starch. Like the starch bacteria will always be, 1,000 times more abundant than the sort of niche fatty acid organism. And because these things vary over many orders of magnitude, I think that's all reflected in the niche size rather than the interaction with immunity. I think for organisms where our algorithm might say, hey, this organism should engraft in the patient, but it doesn't. Maybe those cases, come down to some interaction with the immune system, or they come down to there's already an organism in that niche and it's out competing the one you're trying to put in, and I would suspect both of those answers are true. So you'd have to design an experiment to kind of tease those apart, which I think we should do. But diet studies in human are really tricky.
Yeah, my question has to do with defining a healthy microbiome. So we've done all this work in the past, there's all the consortium in Europe. How translatable is that to other places? I mean, can you find specific texts that define a healthy microbiome or functions?
Yeah, so what we've done is we've looked at all of the kind of case control studies, clinical studies, where they've looked at healthy people and sick people in the same study. And we tried to identify what are the bacteria that are really associated with health. And there are bacteria that are associated with health in more than one disease. There's not a lot of bacteria that are associated with health in particular across all diseases. Now, that's not to say, I mean, there's a lot of bacteria that are quite ubiquitous, and if you got rid of them, you'd probably be unhealthy. But there's not a lot that we've noticed in, looking broadly across many different diseases, organisms that really relate to health. Now, if you look at diseases, you see something a little different. So there's a strong signal for things like colorectal cancer, and in colorectal cancer, what you have is a few bad guys. You have organisms like Fusobacterium that are enriched in cancer patients versus the healthies, and then you have diseases like inflammatory bowel disease, where apart from some patients being enriched in maybe E. coli, their phenotype is different. They're depleted in many healthy organisms, and things like diarrheal diseases like C. diff are sort of a combination of the two. You have a few of these a few overt pathogens, and then you also have a highly changed environment because they're affecting sort of the milieu of the GI tract. So different disease by disease, you see different things, but there's no kind of universal probiotic.